Okay, so I'm just going to identify that today is Monday, June 8th or June 7th, right? So, and I'm here with one of your fellow students, and she asked a question about the short answer section that's coming up on the next lecture quiz, which is lecture quiz 3. It has two components. It has the regular objective portion, the multiple choice, matching, whatever, and then there's also a short answer section, and that short answer section is due the following day so that you have time to kind of you don't have to take both of them in the same day, because I realize that many of you have time limitations, even though this is a summer class. And as I've reiterated several times, you should be spending hours per day on the course. I realize that sometimes you don't have all those hours on the days that, that I'm requesting them. So that short answer section will be due the following day. I can just share screens with you guys real fast to show you that. So I did just move the lecture quiz and lecture quiz 3 and the short answer section to the bottom of module 4 here. Notice there are a lot of connect work for this because I've tried to divide it up into small chunks, mostly because, again, people in the past have complained that when I have everything for chapter 11 all together, the connect takes hours. So I've divided the work for 7, 11, chapter 3, and chapter 12 into small bites as best I can. Again, I would remind you that the connect, this is homework. It's to help your grade. You can almost think of it like extra credit. But it is not, those are not specifically the questions I'm going to ask on the quiz. I think in some of your other courses, from what I'm hearing from students, is the professors use the exact same connect questions on their quiz as were in the homework. And clearly, as you guys have all figured out by now, and as I said in the course outline, that's not what I'm going to do. I'm going to follow, the quizzes are going to be based on the lecture material, on these video lectures that you watch and the notes that you take. So anyway, one of your colleagues, Amber, had a question about the format for these short answer questions, what kinds of questions they would be and what kind of answers I'd be looking for. And of course, rather than giving you examples from chapters 7, 11, and 12, I can think of very quickly on the fly. I'm going to think of some short answer type questions that I, from like chapters 8, 9, and 6. These are examples only. Okay, so I'm going to go switch to a whiteboard here, and very quickly I'm just going to text. So for example, if, well, blue, that's kind of pretty. Yes, if I asked a short answer question on material from the last lecture quiz, which would have been lecture quiz 2. Right, this is the kind of question it would be, and I'm going to go through and real quickly give you the kind of answer that I would be looking for. So the last lecture quiz, you might remember, was chapters 8, chapters 9, and chapter 6. Chapter 8 was all about metabolism. So that particular, as an example for the metabolism, I may have asked compare and contrast aerobic respiration, anaerobic respiration, and fermentation. All right. So oftentimes the short answer questions are compare and contrast, and what that means essentially is this would be like a Bloom's level 3 or 4. In this case, when I ask for compare and contrast, the simplest thing to do always is to start with basic definitions of the terms that are in the question, because that often elucidates what is the same about the things or what is different about the things. So in this example, for compare and contrasting aerobic respiration, anaerobic respiration, fermentation, if I were the student, I would answer this. What do they all have in, oh, I don't like red. I'm so angry, right? So what do they all have in common? I would start usually with something like all 3 of these forms of metabolism. So that tells me that you know that they are about metabolism, right? You could say are regulated by enzymes and provide energy, and in this case I would expect you to be able to say ATP for cells, right? For cellular work or for cells. And then I would expect you, so that's something that they all share in common. And then I would expect you to tell me the things that are different about them. So obviously the aerobic respiration involves an electron transport chain and ATP synthase and presence of oxygen. Anaerobic, and gives you those 38 ATP. Anaerobic respiration does shorter electron transport chain, right? And gives you fewer ATP. Fermentation lacks the Krebs cycle, and there would be no ATP synthase in that case, and you'd only produce 2 ATP. So there's lots of choices for you. I would not expect you to be able to tell me everything about those 3 different types of metabolism, but I would expect you to hit the high points and hit the same high points for all 3. Does that make sense? So if you're going to go after telling me about the final energy production for aerobic versus anaerobic versus fermentation, and you wanted to say something like aerobic respiration produces the most ATP, anaerobic a medium amount, and fermentation the least, then I would expect you to be able to hit those high points the same for all 3 items. And then that would be good, right? That would be enough. It's a short answer question. It's not an essay question, right? I'm not looking for a 5-paragraph essay. I'm looking for, I don't even know that I would say a paragraph, right? Okay. So on this particular question, there's a lot of processing to do to decide, okay, what am I going to use for my points here to show the differences between these 3 things. And so I would expect less writing, but more sort of succinct, clear, focused writing. Now, on the other hand, same set of material, right? And this is what I always ask for the short answer. One of the short answer questions on the last quiz, when we have these in person, right? I always ask, describe the steps of translation. And they're always worth 5 points. And I would expect you to literally regurgitate those 5 steps of translation that were in the notes, in the video that we went over again and again and again, right? The messenger RNA binds to the small subunit of the ribosome. The first transfer RNA binds to the binds to the message. The large subunit binds, repeat, back to step 2 until you hit a stop codon. I would expect, that's what I would expect. And notice it's described, right? So that's more of a level Bloom's 1 and 2. It's like, do you just know this information, right? I put it as a short answer question usually on that particular lecture quiz, because this is a really important thing that you have to know forever now, because translation is how cells make proteins, and proteins, as I'm sure you already know from A&P, right? Most of the hormones, most of the control mechanisms in cells, as well as all the metabolism, are all regulated by proteins. So this is a really important process to understand. I'm trying to think of one from the viruses. I might, from the virus chapter, I probably would have asked, not compare and contrast, but just maybe compare a lytic and lysogenic bacteriophage, paralytic and bacteriophages there, right? So I would expect you to be able to tell me about the lytic cycle and the lysogenic cycle very quickly and simply, again, hitting the high points. Always when it's compare and contrast, that opens it up for you to sort of gives you more liberty with your creativity. I understand on an exam when the time is limited, you don't often have time to think of all of those things, and I totally get that. So what I would say to you is to prepare for short answer questions. Okay. So I'm looking at chapter 7 in my book right now, and I'm going to the back of the chapter. For me, I have a physical copy of the book on page 192. Bloom critical thinking questions, Bloom's level 3, 4, 5, apply, analyze, evaluate. Explain what is happening to bacterial population in the diagram at the top of page 193. Discuss at which point on the graph would be best effective to test the effectiveness of a new antibiotic drug, right? That's a short answer question. While preparing food for the class picnic, Morgan introduces 20 bacterial cells into the pasta salad. So that's going to be basically a question about bacterial growth and how quickly those bacteria can multiply. What is G0, right? It's asking you, okay, she introduces 20 cells, 20 bacterial cells. So G is 20, G0 is 20, generation 1. Those are going to double. So then you're going to have 40. And I didn't even read the whole question, but right, if it asked you, that's the kind of question I would ask. So if you look at those critical thinking questions, those are great ways to prepare for analyzing the material in the chapter. So I'm looking again at chapter 11, chapter 12. For chapters 11 and 12, I would focus on the applications because I'm going to ask you less about, like, for example, in chapter 11, I'm going to ask you less about does dry oven sterilization destroy proteins? Does it damage cell membranes? I'm not going to ask you so much about the mode or how those physical and chemical methods kill bacteria. I'm going to be focusing more on the applications. Like, could you put a mattress in a dry oven? Obviously not. Could you put a mattress in a autoclave? No, you don't have one big enough. So how would you decontaminate a mattress, right? So thinking about what gets done and what's convenient, what is quickest, and what meets the level of sanitizing, degerming, disinfection, or sterilization that is necessary for a particular item. So for those, I would focus on the application. So those short answer questions, I believe how it works is, I'm trying to remember. I'm pretty sure that what happens is you get I think 5 appear and you choose 3 of the 5. It'll say that they're all worth 3 points, but they're actually worth 5 points because it's the only way that I can set it up in Canvas to allow you to see multiple questions and then you make the choice of which 3 you want to answer. So I have blocks of questions, right? So I have batteries of questions or banks of questions, whatever you want to say, for each of those 3 chapters. And I think everybody gets, I don't know, 1 from chapter 3 and 2 from this one and 3 from that one or whatever. So in the each individual quiz will show 5 choices. You choose 3.
And then how much time after you choose those 3?
I think I've set it at 30 minutes. I think I've set it at 30 or 35 minutes. So it's a 1 attempt. So you can only open it once, because if you open it again, you're going to get different questions, right? So it doesn't keep the same questions. So, and everyone's going to get slightly different questions. And, but I feel like 10 minutes is enough for you to read the question, think about what you want to say, and then type, right? Don't spend 15, it's not worth spending 15 minutes on each question. Some of them, like if it's just a describe, then just describe, right? If one looks, it's one, and like I said, you can change your choices as you're going, right? So they're all 5 are going to show up and you're going to go, oh, I don't like that one. I don't like that one. Oh, I'm going to answer this one. So you answer that one and then go back and say, no, okay, that one's hard. I'll do that one last. This one, I think I know a little bit about this one. I'm going to use my notes and figure out what I want to say about that. Try very hard. Don't just copy stuff out of the book, because I'm going to mark it wrong or take points off, right? So everything gets run through Unicheck. And one of the things, because the textbook is loaded, it's checking from how much does this match to the textbook. So don't do that, right? Just put it in your own words. Any word you use, be sure that you define it so that I know that you know what it means, right? So like if you want to say something's a thermophile, you have to tell me that you know what a thermophile is, right? You know, a thermophile is a bacteria that lives at this temperature range. So be sure that you define your terms, because that's how I know that you know what you're talking about, right? Just because you're in microbiology, you can't just start throwing words around that you think I know, you know what you mean, right? No, you have to explain it. That's a good tip for this.
Thank you.
So, and again, that's the next day. So focus on getting the objective stuff out of the way on whatever day that is, probably Tuesday of next week, and then spend a little time preparing. What I would do is I would read all those questions at the end of the chapter and try to think about, oh, would she ask something like this? How would I answer it if she asked something like this? I mean, it's not so important to get to all the nitty-gritty details of answering all those questions. It's more about how would I answer this question, so that you're training your brain for when you see that type of question, how would I answer this question? Right, so it's more another test taking skill that we're trying to teach you on top of all this microbiology. And I don't know about you, but I think these chapters, some parts of them are more boring, like they're boring to lecture about, but they're a little more interesting for the students because it's more applications about, you know, how microbiology is important in hospitals and clinical settings, and how it's different in a veterinary setting compared to a dental setting, compared to a hospital setting or a doctor's office setting. So it does give you a little bit more scope of, oh, maybe that's why they do that. And don't bother to try to learn all the names of the drugs. Go to toxicology or go to pharmacology in nursing school, learn all about it, right? I'm much more interested in that, you know, potentially how a drug will work, because every insert for every antibiotic, that's what you see when you get the drug, right? You open up that insert at the top, it shows a picture of the drug and it says this drug is a cephalosporin, a type 2 cephalosporin. And then you can look up what do cephalosporins do, right? And you're like, oh, those are better for gram negatives because they target cell membrane rather than cell wall, right? So they break up phospholipids. Oh, okay, but they're going to have more side effects. So I'm much more interested in that you remember and understand these are the main ways that drugs can kill bacteria and kill fungus and kill viruses or inhibit viruses than any specific drug, because the drugs are going to change. They change all the time. Sometimes as fast as 6 months they change. So that's why when I think it's in that chapter about the testing of different antibiotics, I think it's in the front of that chapter. I think she moved it from the back to the front. That's why evaluating antibiotics, you have to have the chart that tells you the zone sizes and whether that indicates sensitive, intermediate, or resistant, because those zone sizes can change depending on bacterial resistance, and they actually can be part of the country specific. So if there's an area in the country where a particular drug is being used more frequently, maybe because of cost or because, so let's say it's a poorer region of the country or a community health system rather than a metropolitan health system, they're going to probably be using a little bit different combinations of antibiotics. And so the resistance is going to build up differently in those different areas, right? So then the chart changes. Those charts are updated, I believe, every 6 months, and they reflect that, right? So when you see like the evaluation chart, there's a sample of one, right, in on page 316, and it shows different drugs, right? And then they give you just for, you guys can't see that very well, but they give you an example just for Staphylococcus. And that's because Staphylococcus is particularly difficult to kill. So the numbers, the zones of inhibition need to be larger for staph than they do for E. coli, or than they do for strep, or for this strain of strep. So it's a combination of figuring out what is the bacteria, what strain of the bacteria, as well as which drug will inhibit it best, or either bacteriostatic or bactericidal. That's the beauty of using the broth dilution test, because it actually shows you death rather than just, so bactericidal rather than just bacteriostatic, and you can figure that out in one test. The Kirby-Bauer does not, right? It just shows you whether it inhibits, but you don't know if the bacteria is dead or not. So things like that. What else do I have in this chapter? Blah, blah, blah. Just various things that are highlighted. And then we move to microbe human interactions. That's a little tougher. Again, those of you that are in A&P2, you'll have had a lot of this. Some of the words will be familiar and they'll be used in a slightly different context, primarily because your author of this textbook is actually an immunologist like myself. So we use the same terminology and it's a little bit different than how it was presented in A&P. They don't use the words innate antigen and exogenous antigen the same ways, mostly because, like I said, it's not how immunologists talk. So some of the terminology is slightly different, but you'll see the meanings mostly the same. And then at the end, we'll have a couple chapters of specific diseases, right? And organisms that cause those diseases. Please start soon, if you haven't already, after you finish your lab quiz and your microscope work, be prepared that you're to enter into this second wave of module 2 for the lab kit, right? Read this first, the scheduling document. It's a sample. It's not going to be perfect for you, but if you have it and you kind of plan out how your system is going to go, it will help you. Since it is summertime, I would imagine for almost all of you this, and since it's like 90 degrees this week, it's going to be really hot. Your bacteria are going to grow really quickly. So be aware you may have to make adjustments if the air conditioning where the bacteria is only set at 68 or 72. They're going to grow a lot more slowly than if they're in your kitchen and they're growing at 90 degrees, right? So be aware, I get it. You want the bacteria to be someplace where they're away from your family and your pets, just for, you don't want anybody, mixing up your cultures or, dropping any of your test tubes or knocking something over, right? All of those things, the lab safety things. And you want the cultures to be someplace where they're, and I know it's hard for everyone to have to figure it out for themselves. We're not going to grow too fast. They're not going to grow too slowly. Bear in mind that when I made these videos, right? So these lab day videos that I made, lab day video 2, where's day video 1? Oh, probably here. Those were done in January or February. It was cold and I was doing them in an area of my house that is cold. So I was like, nothing today. I can't do this. Okay, it's 3 days later, not tomorrow. Like I said in the schedule, it's 3 days later and the bacteria have finally grown. So those, don't expect it to be like that, because now that it's hot, the bacteria will grow very quickly for the most part. Some of them will grow faster than others. Right? If ever, for example, once you start growing your E. coli and your staph, if they're growing too quickly, you can always move them to a cooler place while you're waiting. Micrococcus is the one that's the slowest grower of them all. So if you need to wait a day or so because the micrococcus is grown up enough for your experiment that you want to do for the streak plate or whatever it is, keep the micrococcus where it's warm, move the serratia to where it's a little cooler. Right? So you can make those adjustments as well. It's an experiment, right? Try and see what happens. Always go conservative so you can do it again. They've given you enough supplies to almost do most of the labs at least 1.5 times, if not completely twice over. So plan ahead as best you can. Watch the videos first, because like I said, it's me saying, do this, do this, do this in this order. Don't do it like it says in the handout, do it this way. And if you follow those along, you'll get through it fine. But expect to babysit these things every day for lab module 2, right? So
I do have a question.
Yeah.
The microscope and the safety, they're listed as quizzes. Do they count as quizzes or do they count as?
They're in the lab. They're in the lab portion, and they don't count as lecture quizzes, but they count toward your lab grade. Okay. So the lab safety quiz, I don't think is timed, so everyone should get 100.
Either of them.
Yeah. The microscope quiz, I can't remember because I kind of added that just to help you, because at some point you may have to actually use a real microscope in class, maybe in the fall. So to try to help you learn the parts of the microscope for real. So I don't remember if I timed that one or not, but those count toward your lab grade. I'm pretty sure they're both in the, let's check that right now that you mention it, because
I was hopeful that it might boost my my quiz grade.
yeah, no, I don't think it's going to.
Okay.
The lecture quizzes, that's really important. I mean, that's one of the pieces of the course that I have to keep the same. Those lecture quizzes are the basis of your grade, just like in the real course. And you have to score well, or at least 70%. If you're looking to get at least a C, you have to score in that neighborhood to get that grade. And that's true and is going to remain that way. So yes, it looks like if you look at the virtual lab and the lab kits, the safety quiz and the microscope quiz are in with your virtual labs and your microscope. If you do this work and you do it well, and remember for lab report 2, I do need a photograph of you with some of your results so that I know that you're doing this work yourself, not your mom or your brother or your sister who's a doctor, right, and is doing it for you. I need an actual picture of your face, not just your fingernails, right? An actual picture of your face with some of your results for lab report 2. So that'll have 2 components. Usually people upload 2 files for that. Anyway, if you do this work, if you just do it and get it done, it's mostly free points. I mean, there's a few things that I'm looking for, and like I grade exercise 2 through 6 the hardest, but the rest of this stuff, it's just do the work and turn it in, and then you get 100, right? Obviously, these are virtual labs, you know already in Connect, you can't stop doing it until you get 100, basically. So if you just budget enough time and you sit there and you keep doing it over until you get it 100, then you get 100, right? So the lab portion and all the Connect homework, which is a pain, I get it. And you'll notice it drops off significantly after chapter 12. I don't have any Connect for 13, 14, 15, 16, 17, because I want you to focus on the lecture material, because that's what's going to be on the test. I threw some back in for 18 and 19 because these disease chapters, the repetition helps you remember. So I just put them in for the repetition. As I think you've heard me explain on some of the other videos, if you've been watching the lecture videos, inside Connect, I cannot pick the questions. I can only pick the concepts. So that is why I'm not asking you to do a lot of Connect for 13, 14, 15, 16, 17, because I don't want, what I want you to know is not necessarily what the textbook wants you to know. And I can't choose which questions I want you to focus on. So it's different in mastering biology. For those of you that use mastering or have used mastering for bio or chem, bio 1 or chem or A&P, we pick those questions question by question. I read the question, I'm like, that's one, because that's the kind of question I'm going to ask them on the test. In Connect, that's not possible. So I want you for 13, 14, 15, 16, 17, I want you to focus on the material that's in the lecture exam, that's in the lecture videos, right, for the quizzes. All right, so that's enough. All right, I'm going to stop recording.